Damen und Herren, guten Abend und herzlich willkommen zum letzten Vortrag in unserer Vortragsreihe Erbgut editieren. Bevor ich Ihnen unseren Gast heute Abend vorstellen darf, zwei kurze Hinweise in eigener Sache. Am Ende dieses Vortrages wird es unten im Erdgeschoss links ein Apero geben. Der Präsident und Vizepräsident des Collegium generale werden dort verabschiedet und Sie dürfen alle profitieren, indem Sie sich dort am Apero gütlich tun. Der zweite Hinweis ist, dass das Programm im nächsten Jahr, das Thema wird sein, alles falsch, und Sie werden dann ein Programm im Januar zugeschickt kriegen zu dieser hoffentlich sehr spannenden Vortragsreihe. I will now switch to English to make sure that not everything is wrong that I'm telling you now, and let Jonathan fact check my introduction to his person. So we're very happy to have Jonathan Jones with us tonight. Jonathan Jones is a professor at the University of East Anglia, and he works at the Sainsbury laboratory in Norwich, which is a laboratory dedicated to plant microbe interactions. And Professor Jones was already a founding member of that laboratory in the 80s. He's a member of the European Molecular Biology Organization, of the Royal Society, and a foreign member of the US National Academy of Sciences. And one would think that if there would be something like a Nobel Prize in plant sciences, he would certainly be a great candidate for that as well. Professor Jones studied natural sciences in Cambridge, did then a PhD in plant breeding, a postdoc in Boston on symbiotic nitrogen fixation, and then he worked for a private company for a while. It was an agricultural biotechnological company, a company that was the first to actually bring a transgenic crop into the market. So from the beginning, he was very involved in the production of new crop varieties using modern genetic technologies. And he then went back to science, and at Sainsbury, he has been really focusing on plant-pathogen interaction, trying to understand and making tremendous progress in understanding how the plant immune system functions, how plants recognize a pathogen, how pathogens evade that recognition, and how the plant then recognizes the evasion mechanism again to recognize the pathogen anyway. And this is the zigzag model that many of us have grown up with in the plant sciences, that has really found its way into textbooks and is one of the many important contributions of Professor Jones to the field of plant pathogen interactions. What's very interesting is that certainly over the last years, Professor Jones has focused on translating these findings into application by trying to make potatoes resistant to Phytophthora infestans, to blight, which is one of the major oomycete pathogens of potato that caused a great famine in Ireland, for instance, but I'm sure Professor Jones will tell us more about all this in a minute. So we are really happy to have him here because he's an expert in plant pathogen interaction. He bridges the gap between fundamental science and application in agriculture, and he's certainly an expert in modern breeding and genetic technologies for the improvement of crop plants. So without any further ado, Jonathan, thank you very much for being here. We're looking forward to your talk.
Thank you very much. Well, it's an honor to be on this program, and thank you very much for being a generous host. I'm going to talk about how to not be a good host, but Matthias has certainly been a very good host since I got here. It's quite daunting looking at the list of topics here on ethics and editing, and I don't think you're going to hear anything very profound from me about ethics. To the extent I touch on that, it'll be very simplistic. But what I hope to get across to you is the tremendous advances in our understanding how plants resist disease and how pathogens evade resistance mechanisms, and how we, based on this knowledge, we can now have much more sensible approaches to winning the evolutionary game, or at least ending up on the right side of the evolutionary game between plants and pathogens. So, if you look at this field here, this field is a field of wheat somewhere in northern Europe. And what you've got is millions of genetically identical plants. This is completely unnatural. Some people worry about the genetic modification method as being unnatural. Believe me, it's nothing like as unnatural as putting out millions of genetically identical plants in the same field and expecting them to not go down to disease. Because if you've got one race of a pathogen that can grow on one of these guys, it can grow on all of them. And that's why very large amounts of crop protection chemicals are applied. And we'd like to really replace chemistry for controlling plant disease with genetics, because it's a more sustainable way to address the problem. If you're a pathogen looking at a field like this, you're thinking lunch. And I think it's very important to take forward make the most of our knowledge if we really care, instead of just say we care, about the UN Sustainable Development Goals. I guess I am getting into ethics after all. So if you care about zero hunger, good health and well-being, responsible consumption and production, and climate action, what we have to do is to make our agriculture more sustainable, and key to that is reducing the CO2 emissions associated with production. And part of that is trying to not have tractors going up and down all the time emitting CO2, applying agrochemicals, where we could use genetics. And actually, it's going to be a very lively topic next year, because next year is the self-declared International Year of Plant Health. So we'll be hoping to use that to hang some of our work and our public engagement on. So I'm very grateful to so many people coming out, 6:15, when you might be rather doing things with your family. And many of you are probably not experts in this field, so I'm going to try and represent as simply as I can an executive summary of where we've got to in our understanding of plant immunity. And the key point is this: plants have got really, really good defense mechanisms. They can stop pathogens, but they can only stop them if they know they're there. So how do plants know that the pathogens are there? They use receptors. So they've got these powerful defenses, but they are activated upon perception. So there's two kinds of receptors, broadly. There are receptors that sit on the cell surface and detect molecules that sort of leak out of pathogens that they can't help leaking. There's a bacterial protein called flagellum, and there are receptors for that on essentially all plants. And there are other bacterial molecules that only some plants have receptors for. But that activates what's called pattern-triggered immunity, PTI here. And these receptors activate a signaling chain within the cell that activates this defense mechanism. Now, pathogens, to be successful then, have to do something about this. And one of the things they do is they deliver into hosts molecules called effectors. Whether they're bacteria, they've got one kind of a delivery mechanism. Fungi and oomycete pathogens have another kind of delivery mechanism from very specialized structures that project into the host cell. These effectors, their job for the pathogen is to shut down the pattern-triggered immunity. In turn, the third sort of leg of all this is the recognition of effectors by intracellular receptors. So these effectors can be detected by molecules that are, the cartoon version looks like this, and the outcome of that is what's called effector-triggered immunity. So you've got pattern-triggered immunity and effector-triggered immunity. And the classic genetic basis for investigating this has been to try and understand the role of resistance genes, and there's a lot of variation in resistance genes in host plants. And resistance genes usually encode these intracellular receptors rather than cell surface receptors. So this is a bit scientific, but there are three main classes of these intracellular immune receptors that have different flavors of signaling domain at the N-terminus. The details don't really matter. Some of these, they're called NLR proteins because they've got a nucleotide binding site and leucine-rich repeats. This bit of the protein has the job of recognizing molecules, and this bit of the protein has a role in activating signaling. And if you look at the model plant Arabidopsis, you can build a phylogenetic tree of all these different classes, and you learn something about the genetic variation in your model plant. And it turns out that plants and animals have proteins that have this receptor role, intracellular receptor role. Plants have hundreds of them. Many animals have hundreds as well, such as sea anemones and corals, sea urchins. But humans and mice and other mammals actually have relatively few. And that's because we've also got a different kind of immune system, the adaptive immune system, the antibodies that we rely on and that we activate when we have our flu jab every autumn. So they have a similar kind of architecture. They've got a nucleotide binding domain, and they've got a recognition domain and a signaling domain up the N-terminus. And even fungi have similar organized proteins that have receptor detection capacity here, the nucleotide binding domain, and different domains at the N-terminus. So, okay, so how does this work? Well, it turns out that, and a really crucial paper was reported in April of this year, that they work the same way as many animal protein internal receptors work. So there are proteins called, in mammals, APAF1, or in mice, SED4, or in other kinds of proteins, inflammasomes. And what happens is that upon detection, this single protein oligomerizes, and oligomerization means that the N-terminal bit, the front end, you get a so-called induced proximity at the front end, and that bringing these domains together gives you an outcome that was different than what you had to start it with, and you get activation of protease cascades in the case of these proteins, or you bring together protein domains that make holes in membranes and activate a cell death defense response that is part of what restricts the pathogen. So that's, I'm not going to say much more about the mechanism of these NLR proteins, but the biochemical mechanism. I'm going to say a bit about their role in populations. So what I've tried to represent here is something rather like what I showed you on the first slide, a field where everybody's genetically identical. And so the recognition capacity conferred by this resistance protein is the same in every plant. And if it's the same in every plant, then when a pathogen gets into one of these plants, you know it's going to end badly, because everybody is susceptible and the whole field is toast. And that's what you're striving to avoid if you plant a big monoculture. What I've got represented over here in cartoon-like form is each of these colors is meant to represent a different perception capacity, a different recognition capacity. And so if you've got a pathogen that gets in on one of these guys, it's quite a long way to go, or further to go, certainly, to find another plant that's susceptible. And so the epidemic is slower, and the worst that can happen is you only lose 20% of the plants in the population. So the point of this is perhaps sort of Van Gogh's representation of this heterogeneity that's out there in more natural populations or earlier stages of crop domestication. So if you've got a lot of heterogeneity for perception capacity in your field or in your population, then at the population level, you're going to have more resistance. And in fact, by asking our resistance genes and proteins to work in something like this, you're asking them to do more than they've ever been asked to do before. I should say, something I'd like to point out is that if it wasn't for all this disease, we wouldn't have sex. The argument that's made here, and that I think is generally accepted, is that without this constant need to shuffle the deck of a recognition capacity of receptors for parasites and pathogens, then we'd all just reproduce by budding. It'd be pretty boring, but we would. And the reason for that is that it's more advantageous genetically to leave all of your gametes, all of your genes to the next generation by clonal propagation than to just leave half of them, which is what we do when we mate and give rise to progeny in, let's say, what's the more conventional way. So, how do we investigate immune receptor diversity? So we developed a trick that takes advantage of some methods that are often used in sequencing and genomics. In a typical plant, let's say Arabidopsis, actually it's not the most typical of plants because it's got a small genome, but it's got, let's say, 150 megabases of sequence, and it's got like 150 immune receptors. And what you're interested in is variation in the immune receptors. So you don't want to sequence all the rest of the DNA that's there. You only want to sequence the 1% or so that corresponds to the immune receptors. So we developed a method called resistance gene enrichment sequencing. Basically, it's a trick that enables you to just capture the subset of the DNA that encodes immune receptors and sequence that, and not sequence the intervening DNA, the rest of the DNA that doesn't encode immune receptors. We published this quite a long time ago. It's a well-known trick. When you then get a lot of reads from your sequencing runs, you can see the reads stack up over where the resistance genes were and not in between. So it basically just saves a lot of money if you're trying to explore immune receptor diversity. So we've applied this to this wild weed, Arabidopsis, much beloved of plant scientists, at least plant scientists who want to do genetics. And we've collaborated with Detlef Eigel's team and Jeff Dangle's team to investigate the immune receptors of 70 different Arabidopsis accessions using long-read technologies. And what this represents, this is one of many important findings from the paper, but what this represents is that if you sequence one or two Arabidopsis accessions or variants, then you might see 140 to 160 resistance genes. If you see, and what you've got is a cumulative total of how many different kinds of resistance genes you can see. And as you sequence more and more, you see that actually what's called the pan-niblerome, or the pan-NLRome of the species Arabidopsis, there's about 230 different resistance genes. And if you just look in one Arabidopsis accession, you only see about 60% of the diversity of resistance genes that's in there. And we published this earlier this year. Here's the bottom line, or the executive summary. The reference NLRome, I said that Arabidopsis has about 150 NLRs, these intracellular receptors. The species as a whole has 230. So there's tremendous presence-absence variation if you compare different accessions. You'll see resistance genes that are in one that are not in another. And this is very relevant, I think, to the work that we're doing to try and elevate disease resistance in crops by moving in some new immune receptors. Basically, if we put in 3 genes for an immune receptor to elevate resistance to potato late blight, the variation we're introducing is tiny compared to the variation that's already there. Yes, and we've also looked at this in rice. So in rice, there's this region of southwest China called the Yunnan, and these rice terraces, the so-called Yunnan rice terraces. Although all inbreds, all monocultures of a particular genotype, when planted here, are susceptible to a very important disease of rice called rice blast, these farmer land races are resistant. So we thought, I wonder if that's because within these land races, there's actually tremendous variation in the NLR repertoire. So we applied with collaborators this sequence capture method to investigate diversity in the NLRs of rice in these Yunnan terrace genotypes, resistance gene sequencing. And what we found was that within one bag of seed, this land race is called Akuse. This is the depth of this sort of tree here is meant to represent how diverse each individual plant from within a bag of seed is. You can see tremendous heterogeneity within something that's called a land race, more than you even have if you compare 4 different temperate japonica type of rice. And you see this for several of these, not all of them actually, some of them have rather less diversity apparently, but we think that this is consistent with our expectation that the elevated resistance in these bags of seed for these land races is a consequence of substantial immune receptor diversity. So I painted that picture at the beginning of the fields, these monocultures that that are nevertheless that are very vulnerable to a disease epidemic and require all these chemicals to be applied. But so what one could do then, let's say you've got a few resistance gene cloned, you could transform resistance gene A into a particular variety, and also B separately and C separately, and then you plant a mixture and you'd have that mixture. You talk to a company about adopting that strategy and they laugh at you, because if every transgenic event costs $30 million, why would you have 3 transgenic events when you can put them all in the same plant? So it's going to cost you 30 million instead of 90 million, but you don't have that polymorphism. Is there then a way we can think of that would enable us to plant monocultures that actually were not vulnerable to disease? So there's this phenomenon called non-host resistance. So barley I've got 2, this goes over 2 slides. So there's a phenomenon called non-host resistance, and the classic example is barley is resistant to races of wheat rust and wheat powdery mildew, that, and you can plant a monoculture of barley and it doesn't get these wheat diseases, and vice versa. So I think there is if we can understand that well enough, we might be able to find the receptors that would enable us to have durably disease-resistant monocultures. So this goes back to this question of why are non-hosts not good hosts. So most plants actually resist most plant pathogens. So for a pathogen to be able to grow on a plant, it's got to specialize. And so if you think about that process, so wheat and barley had a common ancestor 10, 20 million years ago. And the pathogens that grew on this common ancestor will have diverged to grow on what's now barley and what's now wheat from the pathogen races that grew on the common ancestor 10 or 20 million years ago. So like I said, so barley powdery mildew or striped rust will not grow on wheat and vice versa. And my colleague Matt Moscow at the Sainsbury lab is actually defining the genes in barley. He's mapped to 3 loci that to mean that barley is resistant to wheat strains of striped rust, another very important disease of wheat. So in that sense, this non-host resistance, let's say in barley to wheat pathogens, it's the flip side of specialization. And so then you can ask this mechanistic question, so is this resistance due to the effector-triggered immunity or the pattern-triggered immunity? In other words, the intracellular receptor-triggered immunity or the cell surface receptor-triggered immunity, or both, or perhaps it's something else. So then we got this problem where we like to do genetics on non-host resistance. How do you do that? If everybody's resistant, you can't do genetics. So we were able to develop a trick for our chosen system to study this. It's another pathogen called white rust. It's an oomycete pathogen. And it causes these symptoms. You can see why it's called white rust on brassicacea. You may have seen it on your wildlife walks. It's quite widespread. And there are multiple races of this pathogen, Albugo candida, each of which are specialized on a different brassicaceae host species. So all Arabidopsis accessions resist brassica-infecting race AC2V, which is actually quite an important disease in India of Brassica juncea, which is an important oilseed crop in the drier regions. So the trick is based on this. The technical term is transgressive segregation. So what we guessed was as follows. This is our sort of hypothesis. One Arabidopsis accession, and I've told you how variable they are in their immune receptor repertoires, may resist AC2V because it's got resistance genes A, B, and C. And another Arabidopsis accession is resistant to AC2V because it's got resistance genes D, E, and F. So if you cross those 2 accessions and self, you'll get some segregants genetically which lack A, B, and C, and D, E, and F, and they will be susceptible. Okay? So in other words, transgressive segregation means you've got progeny with a phenotype that's different from either parent. Both parents are resistant, we're looking for a susceptible. It worked, we got a susceptible, we got 2 susceptibles from a big population, and then we could do genetics. We could cross to all the resistant Arabidopsis accessions that we had, which was basically all of them, and chase down the genetics that underpinned this resistance. And the bottom line is this. There are 4 NLR encoding loci to which resistance in Arabidopsis to this brassica-infecting strain maps. And so we can say that the mechanism for non-host resistance in this case is effector-triggered immunity based on recognition of something in the albugo by resistance genes. And furthermore, we can take one of the resistance genes we cloned, WR4B, and we can transform it into brassica juncea, and we can convert it from susceptibility to resistance. So Arabidopsis, this wild weed, is actually probably an infinite source of resistance genes to this important brassica pathogen. There's maybe a slight public acceptance issue of GM crops, but I'll come back to that later. We published this analysis earlier this year in PNAS. So what about applying this to a really, really important disease, not just an important disease? And a really, really important disease such as the late blight pathogen of potato, Phytophthora infestans, the biotic cause of the Irish potato famine, the non-biotic cause, of course, being the fact that the British continued to export corn from Ireland in the 1840s. Back to ethics. But the point really here is that you can go from a field like this to a field like this in about 2 weeks if you don't control it. So it's a completely devastating pathogen. And farmers throughout Northern Europe typically spray 10 to 15 times a year to control the disease. The pathogen's evolving, tolerance of the chemicals that are being applied. It's a $7 to $10 billion a year worldwide problem. So we'd like to be able to turn potato into a non-host for Phytophthora infestans. So we know that we can clone genes that encode these kind of immune receptors from wild relatives of potato, and we did a field trial. This is what I often say is, here's something you don't see every day, a GM field trial in Europe. And this is a resistant potato that's received the gene that we put in, which is called RPI-VNT1, and this is what happens to potatoes in our field trials if they're not protected by having received one of these genes. So this gene's been licensed to the Simplot company, and it's being deployed in the US, and I'm take some satisfaction in the fact that this gene that we clone is the only gene of this class that's being commercialized, at least transgenically. But of course, one resistance gene is not enough. It's too easy for a simple mutation in the pathogen to evade recognition of that effector to overcome. I should say this is December 15th, breaking news. So this lines with this resistance gene and also some tuber improvement properties were finally approved by the EPA and the FDA, in addition to USDA that had already approved it for commercialization. So we've gone to another plant called Solanum americanum to identify additional resistance genes for P. infestans. Resistance to Phytophthora infestans, that's what RPI stands for. So this wild Solanum is actually the diploid ancestor of Solanum nigrum. You may well have seen Solanum nigrum in the UK. We call it black nightshade in sort of paths and on your walks around in the countryside. And it has a lot of helpful genetic properties. It's diploid, it's self-compatible, unlike many of the Solanums. And there's a lot of diversity from seed banks that we've been able to recruit into our program. So we've cloned 2 additional resistance genes, and there's another one that we think we're pretty close to. And what we want to do is deploy combinations of resistance genes, so-called stacks of resistance genes, to attempt to turn potato into a non-host for P. infestans. And of course, it's a slightly religious discussion or definitional discussion if it's really non-host, but I think we'll be able to come up with a very durable resistance with the genes we've already got. But in order to assess how durable your resistance is, you need to know something about the molecule from the pathogen that's being recognized. Because if you know what the pathogen is, the gene what the molecule is in the pathogen that's being recognized, you can then survey all the races at your disposal for whether or not that gene is there. Because if you find half of them are missing that gene, then that resistance gene is going to be pretty easy to overcome, because there'll be some race already out there that doesn't make the molecule that enables their recognition. And there's lots of other reasons for cloning the recognized effector from the pathogen. We're doing a lot of genetics. Our phenotype is resistance. We don't want to clone the same resistance gene a bunch of times, because it's pretty hard work as it is. And you can also, if you've got a lot of resistance genes in a population segregating, you can at least track each one individually if you can track the response to the recognized molecule. And also, if you're trying to make a stack of 3 resistance genes, then you can check that they're all working, if you can check there's a response to all 3 of the recognized effectors. And of course, there's a great biology prize as well, because if you are working with a resistance gene that recognizes a really important molecule for the pathogen, that molecule must be doing something pretty interesting to the host to render it more susceptible. And digging in to figure out what that is interesting. So how do we clone the recognized effectors? So we start with genomics. So this is a nice tweet from my colleague, Sofian. Know your enemy, sequence its genome. So actually, the first genome, reference genome for Phytophthora infestans was reported maybe even 10 years ago. It's been around for a while. And what emerged from early studies on this pathogen, I should say, I've used the word oomycetes. So oomycetes are not fungi. In fact, our community has a T-shirt that says, bats are not birds, whales are not fish, oomycetes are not fungi. Okay. In fact, we humans are more closely related to fungi than oomycetes are. So they're a very distant sort of evolutionary clade. But of course, they cause fungus-like symptoms, and they're often called fungus-like organisms. But they're more related to brown algae. They're brown algae gone bad, okay? If you get late blight in your potatoes, maybe that'll be some comfort. They're brown algae gone bad. So, if we're interested in the effectors, we have to know what we're looking for. And it turns out when the first ones that were recognized by resistance genes were cloned from Phytophthora infestans and some other oomycete pathogens, they all had a bit of protein that enabled them to get out of the oomycete cell, the pathogen cell, and then a bit of sequence that was a motif always associated with the capacity to get into the host cell and do something to it. So this is basically their architecture. They've got a signal peptide at the front that gets them out of the host of the pathogen cell, and then this is associated with them getting into the host cell, and then they've got, quotes, effector domain that does the business to whatever the target is in the host. So you can look at the genome of Phytophthora infestans and say, aha, there are 563 proteins that have a signal peptide and this RXLR motif. And it's probably one of them. Okay, so then you have to sort of sieve through. And basically, if you have a recognition capacity for an effector and recognize the effector, you express them as genes transiently in a plant leaf, then you get a defense response that culminates in death, or a hypersensitive response. So that hypersensitive response is basically, it will enable you to detect if you've got the clone that corresponds to the recognized effector or to some other molecule from the pathogen. So that's what this boils down to. You express your RXLR candidate, one of the 563 with the resistance gene, and if you've got recognition, then you get, the leaf goes brown, you get an HR. So there's various collections from various leading actors in our field. And we screened this lot and it wasn't there, and we screened this lot and it was. So we've got the molecule that's recognized by RPR-AMR3, and it's got some interesting properties which I won't go into in great detail beyond the fact that every race that we've looked at carries this gene. And actually, interestingly, there are a lot of other related Phytophthora species that infect soybeans, that infect strawberries, that infect tobacco, infect palm or cocoa, and so on. And many of them have a homologue of this AVR-AMR3, and indeed their homologue is also recognized by the resistance gene. So this is quite interesting. So Phytophthora parasitica is a disease of tobacco. As I say, we don't particularly want to save the world tobacco industry, but it's still an interesting experiment. And what we found was that the RPI-AMR3 that we cloned from Solanum americanum, you put it into Nicotiana benthamiana, a wild tobacco that's very tractable, and it confers resistance to Phytophthora parasitica. So that's quite interesting. And we've got a tool to test RPI-AMR3 functionality. So what about AMR1, the recognized effector from RPI-AMR1? Was it there? No. Was it there? No. And over here, these are effectors that have not gotten into anybody's libraries yet. So we decided we'd have to figure out which of these are candidates. First thing to do is to find which of them are actually expressed when the pathogen grows on the plant. Which of them is actually, it looks like it's being activated when the pathogen's trying to attack the plant. So we use the sequence capture method. Sometimes people remind me of the observation that to some people, to the guy with a hammer, everything looks like a nail. So we do a lot with capture because it seems to work for us, and that's our hammer that we apply to a lot of different nails. And so we've devised sequence baits instead of to the resistance genes where we do RenSeq. We devised them to all the RXLRs and some other sequences, and we could test then. We've published this method earlier this year. We can test then which of the uncloned RXLRs are expressed and are worth looking at. And basically the bottom line is we found another 50 to check up on, and it was one of them. So this is meant to depict the activation of expression over a time course with various different Phytophthora infestans races. And these are genes that nobody had looked at before. So, so we've got those in our collection, and AVR-AMR1 is one of them. And this just illustrates that hypersensitive response assay. So two different alleles of a resistance gene, AMR1. You co-express with the effector, you get cell death, and you get a weaker cell death with this allele. And with other controls, you don't get anything in a positive control, you get the hypersensitive response. And it turns out this AVR-AMR1 effector class has multiple paralogs, and of course many of the oomycete strains are heterozygous, so there's a lot of allelic variation. But for most of the strains, well, for all of the strains, at least one of the paralogs is highly expressed, and it's recognized by one of the resistance gene alleles that we cloned. And there's an interesting story. I don't want to go into too much detail about this. There's a lot of variation between different Americanum accessions in the sequence of the RPI-AMR1. Like they're only 90% identical to each other. And that correlates with the strength of recognition of different paralogs from different races of the pathogen. But interestingly, again, there are Phytophthora parasitica, Phytophthora cactorum. There are homologs of AVR-AMR1, so that suggests if this is conserved amongst a lot of Phytophthora, it must be a really important effector for Phytophthora. And these forms are very strongly recognized. So back to this question of non-host resistance. So Solanum americanum is spoken of as a non-host for Phytophthora infestans. Basically, it doesn't get Phytophthora infestans, apart from one accession that was susceptible. And so we were lucky we could use that as one of the parents in all our genetics to chase down the gene that underpinned variation for resistance. So these are all those races, accessions. They're called seed packet number. That's what SP stands for. And now we've got the cloned effector for AMR3 and AMR1. We can look through all of these accessions. And nearly all of them have both AVR-AMR1 and AVR-AMR3 recognition. Some have one but not the other, or vice versa. There are a few plants that are resistant that lack either recognition, but nearly always that recognition corresponds, nearly always these resistant plants recognize AMR1 and AMR3 or one or the other, and that's what this is meant to summarize. So what about Solanum nigrum? I mentioned black nightshade. We don't have quite so many accessions of that, but it turns out that black nightshade, which is also supposed to be a non-host for Phytophthora infestans, most of them recognize AVR AMR3 or AVR AMR1 or both. And there's one accession that recognizes neither, and in detached leaf assays at least, it's susceptible. So I think we can say that the non-host resistance of these, of nigrum and americanum, might be mainly determined by these two resistance genes. And I've already showed you another resistance gene, RPI-VNT1, that Simplot's commercializing. So we're pretty hopeful that a stack that's these two plus VNT1 will give very strong resistance. Okay, to summarize this little section, so we've cloned the recognized effectors for these two resistance genes, and they're widespread in Phytophthora infestans races. And I don't want to get into too much detail about this. We've identified the host target for at least one of these, and that will be interesting. So now we're going to sort of take a little step back, because I approach problems in biology as a geneticist. Okay, so I do genetics. I like to develop. I get a kick out of developing genetic tricks that provides new insights and things. So what resistance gene genetics seduces you into thinking is that resistance is either on or off. But actually, there's a lot of quantitativeness to the activation of defense. So I'm not a mathematical biologist, but I did write this. So I talked about pattern-triggered immunity and effector-triggered immunity, and that minus effector-triggered susceptibility. So these are the sort of three components of the zigzag-zig. So there's always, during a real infection, and this is a real infection of potato by late blight, there's what people sometimes speak of as basal resistance. There's some defense being activated even in a susceptible plant. So there's going to be some weak detection of some pathogen molecules by cell surface receptors. That will be being attenuated by the big raft of effectors that attenuate immunity. And some of them might be being weakly recognized. But obviously, in this case, not enough to thwart the pathogen. An important thing to bear in mind is that most resistance genes, they're semi-dominant. What that means is that in a diploid, if you've only got one of the two homologs carrying a functional allele, it's less resistant than if you're homozygous. What that tells you is that the amount of resistance is proportional to the flux through that signal transduction pathway. And if you can double that flux by having twice as many receptors, you'll be twice as resistant. So this is the infamous zigzag-zig. Here representing activation defense by PAMPs. Effectors dragging down that activation of defense, so you have susceptibility. But then if one of these effectors is recognized by a resistance gene, then you get effector-triggered immunity and you stop the pathogen. And there is some pathogen effectors can actually attenuate the effector-triggered immunity, and so it goes on. I like this concept so much I even named my boat after it. Anybody else like sailing? It's zigzag-zig, of course, is what boats have to do to get upwind. So it seemed like a good name for a boat as well. And that's my partner in crime, Jeff Dangle, at the bottom there. So here's another illustration of the quantitative nature of immunity, or the significance of the quantitative nature of immunity. What about if you could just increase this bit? Well, that's what we did. The Arabidopsis EFR gene confers recognition of a bacterial molecule that the solanaceous plants can't perceive. They lack that receptor. So if you take the Arabidopsis EFR gene, which is a cell surface receptor that activates pattern-triggered immunity, and put it into a tomato, then it can recognize that molecule now, and that makes the difference between being susceptible to Ralstonia, bacterial wilt, a very important pathogen in potato and tomato and others of the humid subtropics, and being resistant. And all you've done is increase the amount of PTI. And that could give you that, what looks like a digital output from a quantitative input. Here's another example of this. So this is from work from Dutch colleagues. And if you take a single resistance gene, like VNT1 here, it'll give you like a 45-day delay in sporulation of the pathogen. Here's another one from Chacoense, 30-day delay. If you've got them both, then you've basically got complete immunity. The pathogen never sporulates. So doubling the amount of activation of defense by effector recognition can give you a difference between a sort of quantitative, useful but quantitative resistance and immunity. So if you think about effector-triggered immunity, it's mediated by these intracellular receptors. But if you've got a microbe involved, it's pretty hard to see how you could activate effector-triggered immunity without there having been beforehand some pattern-triggered immunity from the cell surface receptors. Because bacteria are full of PAMPs, this pathogen-associated molecular patterns that are recognized by these cell surface receptors. So how do you study effector-triggered immunity alone? And the answer is we used a genetic trick again. So we set up an estradiol inducible, basically an inducible effector-triggered immunity by taking a Arabidopsis, working with that carries an immune receptor pair called RPS4 and RS1, and that recognizes AVR RPS4. So we had estradiol inducible AVR RPS4, so we can add this compound, and the AVR RPS4 gene is induced, and then the recognition mediated by this immune receptor plays out. And you can see that over a time course from adding estradiol, you get, after a couple of hours, you can see the gene for the AVR RPS4, and then you see a bunch of genes that are involved in making a plant hormone called salicylic acid or piperacolic acid, and that comes up a couple of hours after AVR RPS4 shows up, and then you get PR1, which is a classic marker for late stages of the immune response in response to salicylic acid. So this is the real, the main thing that this ETI does is activate induction of a set of genes that are involved in defense hormone production. So great, we thought, now we've got this method where we can study the hypersensitive response in a really clean way. And so if you don't add estradiol, there's no HR. If you add estradiol, there's also no HR. What? We were a bit perturbed when we first saw this. And then we thought maybe for this hypersensitive response, you need to have both PTI and ETI. And guess what? That's what we found. So these are Pseudomonas or Pseudomonas-derived molecules that are an abundant source of the molecules recognized by the cell surface receptors, the pattern-triggered immunity triggering effectors. And if you've got PTI and ETI, then you get cell death. But if you have PTI alone, then you don't get cell death. If you have ETI alone, you don't get cell death. So they actually mutually potentiate each other. And I'm going to spare you the detail of a lot of protein assays, gene assays. But there are a number of hallmarks of this cell surface receptor triggered immunity. You activate protein kinases, you make active oxygen species that contribute to defense. You make cell wall fortification that keeps the cells more resistant to progress of the pathogen. So when you activate PTI by adding these recognized molecules, then you activate things like MAP kinases and respiratory burst oxidase homologs to make reactive oxygen species. If you turn on ETI, you actually induce genes that encode a bunch of components that are required for PTI signaling, but you don't activate PTI signaling. You enable or you potentiate PTI signaling because there's a whole lot more of the components that are involved in PTI signaling, but you don't activate it. So you just got a lot more of these components. But if you got both, then you get, when the PTI activates a much stronger response, because there's a whole bunch of components there that are elevated levels compared to if you haven't triggered ETI. And I'll just say that actually it's MAP kinases that are most crucial to the HR, because if you put yourself in a position where you can specifically inhibit MAP kinases, the hypersensitive response goes away. And this whole part of the narrative, I should say, is conducted by a brilliant PhD student, Bruno Ngu, co-supervised by a great postdoc, Ping Tao Ding. So we originally set this up as I used to call it the Z scheme for plant immunity. So the Z scheme refers to photosynthesis. So you've got photosystem 1, photosystem 2, and in between you get some decay of the energy of the electrons, PS1, decay, PS2. That's called the Z scheme describing photosynthesis. And that's how that all started. But the way we draw it implies that the PTI and ETI are actually doing the exact same thing, just with ETI you've got a bit more of it. And it's actually not quite like that. So like I said, they interact, ETI induces the components, but they don't activate the components. So now we draw it slightly differently. So here's the pattern-triggered immunity that gives you resistance and response to detecting molecules at the cell surface by these pathogens. And of course, effectors are made by the pathogen to suppress that activation of PTI. But if the effectors are recognized by these intracellular receptors, then what they do is to restore the restore the attenuated pattern-triggered immunity. But it's PTI that's actually stopping the pathogen. And if you've got more of these, you can imagine you've got stronger response. So to recapitulate, ETI induces genes that encode components required for PTI. Just a little anecdote. I mean, many of the effectors that have been defined, most of the effectors whose function has been defined, they impair the action of many of these components, such as MAP kinases or this protein kinase or this protein kinase. So, in some ways, if you have effector-triggered immunity alone, does it activate any defense at all? Well, it does activate hormones that potentiate defense elsewhere in the plant and also locally. So back to this issue of stacks, if you've got multiple resistance genes, there's going to be a physiological benefit as well as a genetic benefit. So not only if you've got 3 resistance genes, is it much harder for the pathogen to simultaneously mutate and overcome all of them, in one fell swoop, but also the amount of defense that gets activated is much higher, and you get really, really strong resistance. And I think this is actually what underpins non-host resistance. And so it's not a fantasy to think that we can reach into genes from wild relatives of our crops and pull out genes that enable them to be non-hosts for the crop's pathogens. So I mentioned field trials. So this is now last year's field trial. We have a 3 resistance gene stack here. It's just completely immune to late blight. Camille cloned RPIMR1 and 3. Marina was involved in the taking forward essentially a product that carries VNT1 AMR3 and AMR1. There's no yield penalty. We actually did a yield trial this year on these lines, and there's no yield hit at all. The yield is indistinguishable from the starting plant material. The industrial partners in this, and we want to add potato virus Y resistance and also this EFR gene to this stack. And this would be very valuable in Highland Africa, where they get, they plant nearly 1 million hectares of potatoes up and down East Africa, and they have terrible problems with blight, PVY, and Ralstonia. So I make the point that we're putting in some resistance genes. As I told you earlier, Arabidopsis only carries 60% of the resistance genes that are in the pan-niblerone of the whole species. So what we're doing is, by putting in some resistance genes, is a tiny change compared to the variation that's already there. I like to say the variation we introduce with the GM method is a pimple on the buttock of the variation that is already there. Yeah, so and interestingly, these lines that have the stack of resistance genes is completely resistant to tuber blight, which is a major storage problem in potatoes. And so, yeah, so this is a uninoculated and inoculated wild type, natural Maris piper potatoes. And here's our stack potatoes, basically completely immune to tuber blight. And of course, now we get into perhaps ethical territory, or in fact, given we're talking about Boris Johnson, unethical territory. But in his first speech as Prime Minister, I was driving down to Cornwall on holiday and I nearly fell out of my car when he said this. Let's start now to liberate the UK's extraordinary bioscience sector from anti-genetic modification rules. Let's develop the blight-resistant crops that will feed the world. So I spent the last 3.5 years trying to fight Brexit, trying to stop it happening. I've been on every demonstration against it. It's been driving me round the twist. But it looks like it's happening. So never waste a good crisis. The tiny silver lining around that big dark cloud is the possibility of setting a science-based regulatory framework, and I will, of course, attempt to take advantage of that. I just want to make a point about the discussion on the GM topic. Of course, GM is not a magic bullet for anything. But in the same way as back in the, probably 10 years ago now, when people talked about addressing the energy security challenge and minimizing CO2 emissions, you'd have arguments between people about whether or not it was better to just insulate our buildings or go switch to photovoltaics or wind or nuclear. And the real point of this wedge concept is let's not have silly arguments like that. We need all of them. And in the same way, for food security, I would argue that to address the huge food security challenge of the next 25 years, up to post 2050, when there'll be a couple of billion more people, many of whom will think that they want to eat more meat. We need to have all of these wedges in place. And we absolutely could have the scope for better agronomy, reducing waste, a lot of benefit from reducing meat consumption, health-wise and otherwise. We need plant breeding, but we also need the editing and using the GM method to address the challenges that we undoubtedly face. So let's avoid false antitheses between one or another of these solutions, all of which are required. So, okay, now to just sort of wrap up. Why are plants not very good hosts for many pathogens? They've got heterogeneity in their immune receptor repertoires. Non-host resistance, I think I'm becoming more convinced, actually it can be mostly explained by immune receptor stacks that confer recognition that is absent from a crop. and if we define what those receptors are, we could help convert the crop to being a non-host. These intracellular and extracellular receptors, there's tremendous variation in crop relatives. It's a rich potential source of new resistance genes. Moving resistance genes from one plant to another is the way to address many crop diseases. But we absolutely need to be able to use the GM method to do that. You won't be able to do that by editing. And these are more sciencey points, but I think it's quite. I would take satisfaction from how the basic science we do on the immune receptor mechanisms and the mutual potentiation of cell surface and intracellular receptor triggered immunity feeds in to our attempts to address a major disease problem in the field. And here are team members who've made a huge contribution to the work that I've told you about. I'd like to thank Gatsby Foundation, which has been putting money into the Sainsbury lab for the 31 years I've been there, and various other sources. I'd like to thank various collaborators and my wonderful project leader colleagues. And thank you for your attention.
Thank you very much for this wonderful talk. Wir machen 2 Minuten Pause, damit die Personen, die sich nicht an der Fragerunde